won't editorialize on the mic here. Okay, so the topic for today, um, we're going to talk about energetics of chemical reactions and kinetics of enzyme catalyzed reactions. Okay, so keep those two concepts in separate parts of your brain. There's some easy to get them intermingled, but they're really quite separate things. They're both very important within our body. I'm going to illustrate these concepts with some specific examples of specific reactions. If you want to study the details of those reactions and memorize them, you're free to do so, but because you will see them in lectures coming up, but really that's not what this lecture is about, right? Those are just examples meant to illustrate these general principles of energetics and enzyme kinetics. Is that fair? Okay. So yeah, we have to do a little bit of preliminary stuff before we can get into the actual fuel metabolism and inborn errors of metabolism and all that fun stuff. I think this is the last one though. So, okay, should we get started? Anybody like the onion? Okay. I'm not going to go through this, but maybe after you go through this lecture, read through this, and it's kind of whoever wrote it knows a little bit about biochemistry. So, okay, should we do a little warm-up quiz? Just a few easy questions here. Yes. 537598. You got it? Okay. Oops. Okay, is that working now? Okay. Okay, so this is harkening back to last week's material. And good, serene's the correct answer there. Okay, now previewing some of today's material. Wow, okay. Okay, so best answer is the equilibrium constant. Okay, so the equilibrium constant, that is the ratio of products and substrates of a reaction when there's no free energy change in the reaction. Okay, when there's a stable system, essentially. Okay, the other ones listed there. Well, I'm not going to tell you what they are before we go to this next this next one. And this one you could kind of guess with the kinetics, and velocity sounds kind of like something that's related to kinetics, right? Okay, so best answer here is maximum velocity. Okay, so again, as I mentioned before, keep these two principles kind of in separate boxes, separate but related boxes, energetic properties and kinetic properties of reactions. Okay? All right, so bioenergetics, this is referring essentially to changes in the potential energy of the substrates of a reaction versus the potential chemical energy and the chemical bonds of the products of a reaction. Okay, and the energetics drives reactions, if you will. That dissipation of energy out into the universe is a driving force for any chemical reaction. So any reaction that happens that has a negative free energy change. Kinetics, this is referring to the speed at which chemical reactions happens. And generally, the kinetics that we'll talk about, these are referring to enzyme catalyzed reactions. So the free energy changes of reactions, that's irrespective of whether the reaction is catalyzed by an enzyme or not. And the free energy change is not related to how fast the reaction happens. Okay, so we're going to be illustrating these examples, these principles with some examples of fuel metabolism processes. And from an energetic standpoint, what we're talking about here is taking the chemical bond energy in a food substrate like glucose and converting that chemical bond energy, the carbon-carbon bonds, into carbon-oxygen bonds to make carbon dioxide. And the potential energy of these carbon-oxygen bonds are less than the potential energy of the carbon-carbon bonds in the carbohydrate substrate. So that energy then is driving the reaction, and within our body we're able to capture some of that energy to do useful things. Okay, so I'm going to go through this energetic discussion somewhat quickly because I think it's probably, the details are not so important, and I think you've probably studied the details before. So focus on the big principles. Okay, if we have a food substrate, such as a donut. The caloric value in a donut, it's got some sugar, but probably most of the calories are coming from fat, like palmitate as shown here. Okay, so we could burn this donut. We could put a lighter on it, and eventually it would have a self-sustaining reaction that would give off heat as the carbon-carbon bonds are replaced with carbon-oxygen bonds into carbon dioxide. Right? Okay, so why doesn't the donut just spontaneously combust if you set it on the desk here? You need to put the lighter on it, right? You need to apply some activation energy. Okay, so this diagram off on the left is showing the potential energy, the relative potential energy of the chemical bonds, the sum of all the chemical bonds in that fat substrate versus the chemical bonds in the 16 molecules of carbon dioxide that would be generated if you completely oxidize a molecule of palmitate. Okay, so the product has more potential energy than the substrate has more potential energy than the products, so this is energetically favorable. Okay, however, it's not a spontaneous reaction because there's this activation energy that you have to overcome in order to get the self-sustaining reaction. So that activation energy is represented here as the difference between the chemical bond energy in the substrates and the maximum amount of energy during the reaction. So the maximum amount of the potential energy during the reaction, this is like when all the bond angles are in your substrate are at their like maximum bent strain before they're just about to break. Okay, that's kind of how I interpret that. Okay, so if we apply the activation energy, that donut will burn, and the burning is going to give off heat, and that heat is going to represent the difference between the chemical bond energy in the substrates and the products. Okay, and that heat, in the context of burning something, like a flame, it's hard to capture that heat efficiently. But we can capture that energy more efficiently when we burn things within our body. Okay, so the delta G, in this case, the delta G, or the total energy change from the starting conditions to the ending conditions of any system, that's the Gibbs free energy change. So that's going to be a parameter of a reaction that is going to be dependent on temperature and substrate and product concentration. And I'll elaborate on that in a little bit. But basically, what's driving the reactions are either going to be changes in heat. So reactions are either going to give off heat or take in heat, and they can be driven by giving off heat. They can also be driven by changes in entropy, and entropy is this tendency towards disorderedness, if you will. Okay, so the universe wants to be this uniform system where there's equal concentrations of everything around everywhere within the universe, right? I think that's one way to say it, right? So if you think of like individual amino acids in a water in a test tube, if they're individual amino acids, they have a lot of different degrees of freedom, and they can diffuse evenly throughout that system. If you took those same amino acids and translated them into a protein, that would have a lot more order to it. Okay, so that is something that is anti-entropic, or that the universe doesn't like energetically, so we have to expend energy in the process of translation, right? Okay, so just an example of that. So you can drive chemical reactions either through changes in heat or through changes in entropy, and the Gibbs free energy equation is shown here, shows the relationship between those. So you won't be asked to solve any problems with this equation, but just kind of get the general principles of it. Now, the equation as it's written here, this is a general equation that can be applied to any chemical reaction. Okay, and if you know the parameters of the reaction, you can describe it with this formula. Now, there's a specific set of conditions that are standardized that we call the delta G0 condition, or excuse me, the delta G0 prime conditions. And the delta G0 prime, this is where we measure the energy change of a reaction under very specific conditions of equal concentrations of the product and the substrate. Okay, so when we start the reaction, if we're going to measure the delta G0 prime, we start it not only with substrate in the test tube, but we also have an equal concentration of the product. Okay, and then if the reaction is energetically favorable, then it's going to have an increase in the concentration of the product. So the delta G0 prime, this, in addition to the equal concentrations of product and substrate, we also have uniform pH and uniform temperature conditions for the reactions. Okay. Okay, so yeah, if the delta G is negative, then you're going to get a reverse reaction. And if the delta G, if the delta G is negative, you're going to get a forward reaction, as shown here. And if the delta G is positive, then you would go backwards. Okay, so that's, I'll provide an example of that in a moment. Delta G's are additive for sequential reactions, and that has some important contexts or important implications for fuel oxidation generally. And then as we go into the kinetics, as we talk about the enzyme catalyzed reactions in more detail, the ability of enzymes to link reactions together has some implications for energetics. Okay, so let's look at our sitting around the campfire and throwing the donut on the campfire and watching it burn. So that's the energy changes depicted here. Or we could bite the donut and burn the fat in that donut within our bodies and do the same, have the same substrates and the same products and the same net free energy change. However, rather than having it done in this rather difficult to decipher chemically process of flame and combustion, we're taking that chemical bond energy and the carbon-carbon bonds of our substrate and we're converting it into carbon dioxide in discrete little steps, one after another. Okay, and each of those steps has its own activation energy, and each of those steps has its own delta G, and they're all additive. And if you they're additive, and the sum is going to be the same as if you burned the donut in the campfire. Okay. However, because the reactions are occurring in discrete steps, we can, in a more regulated fashion, take the potential energy of each step and capture it to do useful work, either mechanical work or biosynthetic work or transport work or what have you. And just as an example of this, so what we're doing, we're taking electrons that are shared between two carbons, making carbon-carbon double bonds or carbon-oxygen bonds, and then transferring those electrons to redox cofactors that we can then oxidize on the electron transport chain and use that to make ATP. Okay. Okay, so each of these little steps has a delta G, and the delta Gs are additive and they can do work. Okay. All right. I guess I should mention here, we are warm-blooded organisms, right? So some of this energy is dissipated in the universe, if you will, and that makes us warm, right? So it's not all about mechanical work or transport work. Okay, so let's look at an example to illustrate some energetic principles. So let's consider a biochemical reaction that is important for glycogen synthesis. That is, so glycogen is the stored form of carbohydrate energy within our body. It's a big polymer of glucose. So the reaction that will convert glucose 6-phosphate into glucose 1-phosphate, that essentially commits that molecule of glucose towards glycogen synthesis and towards the storage pathway. There's a few other metabolic fates of it, but just consider this as committing it to glycogen. Now, the reverse reaction is essential for liberating glucose from that stored glycogen. Okay, if we have glycogen over here, in order to use that stored fuel, we have to do the reverse of this reaction to make glucose 6-phosphate, and then glucose 6-phosphate is a glycolytic substrate and can be used to generate ATP. Okay, so this reaction, if we consider the conversion of glucose 6-phosphate to glucose 1-phosphate, that has a positive delta G0 prime. Okay, now this is a reversible reaction, as I mentioned. So if we go this way, the delta G0 prime is positive. If we go this way, it's negative. It's the same value, it's just the inverse of it. Okay, and consider the delta G0 prime. That is representing the energetic changes when we have equal concentrations of glucose 6-phosphate and glucose 1-phosphate within our test tube. Okay, 1 mole of each within our test tube. Clear? Okay, so little clicker question here. If we had that starting condition of 1 mole of glucose 6-phosphate and 1 mole of glucose 1-phosphate in a test tube, what would happen to that reaction given that we know what the delta G0 prime is? Good. So the concentration of glucose 6-phosphate will increase. Okay, so you have a positive delta G 0 prime for this direction. So that means if you have equal concentrations of them in a test tube, this one is going to accumulate, right? Is that clear? Okay, so the question then is, how do we synthesize glycogen within our body then? This is an energetically unfavorable reaction, right? Yet we all have glycogen in a lot of it in our liver and a little bit of it in our muscle and other tissue. Any ideas? Yes.
Use energy to create the glycogen.
Yes, that is correct. We do. And okay, so I like that answer. That is a good answer. There's a kind of a simpler explanation. Do you have an idea?
Enzymes used to lower the activation energy and make it favorable as a catalyst.
There's a simpler answer that I'm looking for. Okay, what are the delta G 0 prime conditions?
That they equal each other.
Right, equal concentrations of substrate and product, and 25 degrees and pH of 7, right? Okay, is that likely, are those conditions likely to be present within our body? No. Right, so delta G 0 prime, it's a useful measurement to compare one reaction to the other. However, those are contrived conditions that don't generally exist within our body. Okay? So how do we synthesize glycogen? Well, okay, so we've got a positive delta G 0 prime for this reaction, right? Now, irrespective of any enzymes, what would happen to the energetics if we decrease the concentration of glucose 1-phosphate? Kind of intuitively, it would make them more favorable for that forward reaction, right? You're sucking away the product, so that kind of seems like it would make that activation or that energetic hill a little steeper. Yes?
Are you describing the Chatlayer's principle?
Yes, I was going to do it in the context of the Gibbs free energy equation, but I think that Le Chatelier's principle is the principle, and then this is how you would quantify it, or the how you would describe it mathematically. Am I wrong on that though? It's been a long time since I've been in a chemistry classroom, but I guess this is technically a chemistry classroom at the moment. Okay, so all right, you've seen this before, everybody? Let's just focus on the equilibrium constant, I guess, right now. So the equilibrium constant is, if we went back to this situation, if we had equal concentrations of glucose 1-phosphate and glucose 6-phosphate in the test tube, we let nature happen, what is going to be the final concentration of glucose 6-phosphate and glucose 1-phosphate? Right, when all the reaction has happened and there's no more energy change happen. So that's the definition of the equilibrium constant, is the relative concentrations of product and substrate when there's no energy change within the system. Okay, so if we know what the delta G 0 prime is, and the delta G 0 prime condition is going to be equal concentration of product and substrate, then we can deduce or calculate what the actual delta G is going to be for conditions where we don't have equal concentrations of product and substrate. Is that clear? Okay, so let's just look at what would happen if, for this hypothetical reaction, if we decrease the concentration of the product. Okay, there was one other. Okay. When you're at equilibrium, the delta G is 0, right? So if this is 0, then that's how you get that equation. Okay, so looking at that equation, what happens if we decrease the product concentration? okay, that term becomes smaller. Then the log of that term becomes more negative. Then the gas constant and the temperature function become more negative. Then the whole term becomes more negative, and essentially the reaction becomes energetically more favorable. And if you decrease that product concentration, past the point where the delta G flips from positive to negative, then you can turn that reaction into something that is energetically going to happen. Is that clear? Okay, so with respect to what happens within the body, we don't exist in delta G zero prime conditions. Okay, the actual delta G is going to be favorable because we have subsequent reactions in their body that are going to decrease the concentration of the product of that reaction. Okay, and ultimately those reactions are going to generate glycogen. Okay. So each of those reactions has a negative delta G, and that's energetically pulling that unfavorable reaction and making it favorable. Does that make sense? Okay, so, and again, so this was our phosphoglucomutase step, the conversion of glucose 6-phosphate to glucose 1-phosphate. As shown here, this is energetically unfavorable. Now this would be a delta G zero prime unfavorable, right? It's made favorable because we have these other reactions that are energetically favorable that have negative delta Gs, and that's decreasing the concentration of glucose 1-phosphate, and it's essentially flipping this and making it green too. Is that clear? Okay. Okay, so if you're interested in the Stanford pathway, can you see it? It's somewhere right over there. So it's the link between glycolysis and glycogen metabolism. Okay. Okay, should we look at one other example? HMG-CoA synthase. This is one of the steps in converting acetyl-CoA into cholesterol within our body. So we can either eat cholesterol or we can synthesize it de novo. And the HMG-CoA synthase, this is not the key regulated step, but it's one step in front of that. And it's a reversible reaction. So why don't we look at that question? Now, one thing you have to consider, the beta-hydroxy beta-methylgluteryl-CoA, that's like, that's the main product, if you will, of the pathway. But the coenzyme A, that's another product as well. So you look at a pathway and it'll be like, okay, what are you making that the body is going to use for something else? And then there's like redox cofactors or transport cofactors or whatever that are in a smaller font as though they're less important. But really for energetics, it can be driven by changes in concentration of stuff in the larger font and also in the smaller font. Okay, is that clear? So sometimes there'll be a redox cofactor, and consider that just a product and a substrate of the reaction, just like whatever is being oxidized and reduced. Okay. I guess the chart isn't coming up here, but I think decreased concentration of CoA was the best answer here. Right. Which one? Oh, I'm sorry. Looking at the question, it's looking at which would energetically favor that way. So, okay, I'm sorry. So yeah, that would be the increased concentration of HMG-CoA. Okay. Any questions on this basic energetic stuff? Okay, let's switch to talking about enzyme catalyzed reactions, and I'll have a little bit to say about how enzymes affect energetics, and then we'll segue into our discussion of kinetics. Okay. Does this look familiar to anyone? Did everybody do organic lab? Okay, so what do we have there? We've got hot plates. It's in glass because it's probably super corrosive, right? And you can see that like the metal surfaces here are all corroded, right? Okay. So when we synthesize things in the organic lab, in order to get a product, we have to, usually have extreme pH, extreme heat, and what was the highest efficiency of product formation that anyone got in an organic synthesis lab? Did you have to calculate that? What was a good one for your class? 20%? 50%? That was pretty good. Right. So the product formation is pretty inefficient, and you're making your, intended product, but you're also making other things that you generally don't want and you have to purify away, right? Okay, and the reactions occur, the activation energy for these reactions is overcome just by random collision of molecules within that test tube, right? And if they collide with the right velocity at the right angle, then you're going to get a reaction that happens. Okay, so let's contrast that with chemical synthesis in the body. We have a nice, comfortable temperature, right? Neutral pH generally, although there are different pHs in different compartments of our body. We never, ever get unintended products. Well, I think that's fair to say that, right? So phosphoglucomutase, that's going to either make glucose 6-phosphate or glucose 1-phosphate. It's never going to make ribose 5-phosphate or galactose phosphate, okay? It's only going to make its intended product, not only the intended product, but like the actual stereo-specific product as well, right? And the key difference there is that the reactions don't depend on random collision of molecules within a test tube, okay? In the body, most reactions are enzyme catalyzed. Okay, so let's look at energetics of enzyme catalyzed reactions. So what an enzyme is doing is really, it's obviating the need for that random collision of molecules to initiate a reaction. Okay, so the enzyme is holding on to the substrates of a reaction in the right orientation, and it's holding on to the specific substrates and putting them in the right place so that they can click together and make make activation energy that is sufficient for the reaction to happen. Okay, so if we look at a reaction where you have molecules in solution in a test tube and no enzyme present, and they have to just, by chance, collide with one another at the right energy and the right angles to form a product. Okay, so if we have substrates A and B and they're making products C and D, there's going to be this intermediate phase where they're joined, right? If you have, okay, A and B come together to make C and D, there's this point here. Like this is going to be the maximum strained point and the maximum potential energy of the system when you've got bond angles that are strained to the maximum amount before they break. Okay, and we call this the transition complex. And again, if the only way you can get that transition complex and get beyond it in a test tube is through random collision of the molecules. Okay. In an enzyme catalyzed reaction, it's a little bit different. So here you have the A and B, the same reaction, A plus B making C plus D. However, it's not just A and B colliding that provides the energy. You've got A binding to an enzyme and then B binding to an enzyme. So this transition complex has a third component, if you will, the enzyme, that is holding the two products in just the right orientation for the reaction to occur. So the energy of that transition complex is much lower. That means that there's a lower activation energy required for enzyme catalyzed reactions versus non-enzyme catalyzed reactions. Okay, so that's one of the key things that a enzyme catalyzed reaction does energetically versus the same reaction that just occurs in a test tube in the absence of any enzyme. If you look at the delta G, however, the delta G is unchanged whether there's an enzyme present or not. Okay, so if you're just looking at the chemical bond energies in these versus these, it doesn't matter if there's an enzyme there or not. Clear? Questions? Okay, I gotta catch my breath here. Okay, let's look at an example. And the example that I'll use is hexokinase. And there's a related enzyme called glucokinase that catalyzes the same reaction. So shown here, this is glycolysis pathway within our body. Converting one molecule of glucose into 2 molecules of pyruvate. The first step in glycolysis is adding a phosphate from ATP to glucose to make glucose 6-phosphate, and then another product is adenosine diphosphate. Okay, so focus on this reaction. And it's just, well, there it is on the Stanford pathway. Okay, so if we look at that reaction and split it into its constituent parts, if we had a molecule of glucose and inorganic phosphate in solution, and we calculate the delta G0 prime of the generation of glucose 6-phosphate, that has a positive delta G0 prime. Okay, so if we just mix glucose and phosphates in a test tube, it's not going to make glucose 6-phosphate. Okay, so what's the other part of the reaction, though? It's not just inorganic phosphate is not one of the substrates of the hexokinase enzyme. Right, the substrate is ATP. So the hexokinase enzyme is able to couple an energetically favorable reaction, the hydrolysis of phosphate from ATP, so that's a strongly energetically favorable reaction, right? 7.3 kilocalories per mole. So the negative delta G of this reaction overcomes the positive delta G of this reaction. Okay, so that's one key thing that enzymes can do, is to link reactions together, and because delta G's are additive for reactions, the total delta G is going to be the sum of them. Okay, so how the hexokinase does that, when we talked about protein structure, I think I mentioned a lot of enzymes and particularly the monomeric enzymes like hexokinase that are soluble, they have like a Pac-Man type shape, right? It's, roughly a globular globular enzyme. But then there's this cleft that is the active site. Okay, so the way that hexokinase makes this reaction happens and links the phosphorylation of glucose with the hydrolysis of ATP is to bind both of the substrates and put them right in the right place. So if you had hexokinase shown here, and then glucose and ATP in a reaction, first the binding of glucose into that catalytic cleft, that changes the entire conformation of the enzyme, okay, and it kind of closes up the mouth of the Pac-Man, if you will. When it does that, when it kind of closes this mouth, it then creates an ATP binding site that is, and you know, my PowerPoint art skills aren't great here, but the ATP binding site is putting that phosphate of the ATP right at the number 6 carbon of the glucose and allowing that reaction to happen. Okay. So if you look at the energetics, the hydrolysis of ATP is more than sufficient to overcome the negative energy barrier of the adding the phosphate to the glucose to make glucose 6-phosphate. Okay, pretty simple principle. Okay, so let's talk about, oh, and I guess one other thing I'm going to mention here. These binding sites, not only do they provide the linking of one reaction to the other that helps with energetics, they also provide specificity. So hexokinase, you know, it really, really prefers glucose as a substrate, okay? And really prefers ATP as a substrate. It's not going to use GTP to hydrolyze, right? The amino acid side chains here, they're making a, you know, the old model of the lock and key, the substrate being the key and the enzyme being the lock, it's really only one thing fits in there. There are some enzymes that are promiscuous and can use multiple substrates, but generally they're going to be binding a similar similar chemical moiety that's shared by all those substrates. Okay. So enzymes provide specificity of product formation. And I'm not sure if I mentioned that anywhere else. Okay, last chance to ask about energetics before we switch to kinetics. Okay, so before we leave energetics, I guess, how fast does this reaction happen? Super fast, really fast. If there's an enzyme there, it would happen pretty fast. All right, enzymes can speed up the rates of reaction. Now, if you just look at energetics, like, let's go back here, like ATP hydrolysis, it's a negative 7.3 kilocalories per mole. If you just have ATP in water at your bench, it will hydrolyze spontaneously, but not very fast, okay? You can have ATP, you know, pretty stable in solution. So even though it's energetically favorable, that doesn't mean that it's going to be, you know, happening at a rate that is useful for the body, okay? And that's where enzymes come in. And enzymes, we know that they don't change the overall delta G of the reaction, but they do have a very significant effect on the velocity of product formation, the reaction rates. And I guess further to that point, enzymes have an important ability to regulate the reaction rates. Okay, so enzymes can be turned on and off in different circumstances, for example, by phosphorylation or post-translational modifications. Okay, so if we have a hypothetical reaction again, and I'll first do hypothetical and then we'll illustrate it with some examples. If we have a substrate S that makes a product P, if this is an enzyme catalyzed reaction, first thing that's going to happen, your substrate has to bind to the enzyme to make that enzyme substrate complex, and then that's going to have that transition state where you get the maximally strained bond angles, then the reaction is going to happen, and then the product is generated, and that diffuses away from the enzyme. And typically the enzyme is either unchanged during this reaction, or if it is changed, there's some other side reaction that will regenerate the enzyme. Okay, so let's look at, and this is not anything to do with the energetics of the reaction. Okay, let's presume that it's a negative delta G and that it's going forward. Okay, so if the reaction is going as shown here, the velocity of that reaction is going to depend on primarily on the relative concentrations of the substrates, and if there's more than one substrate, it'll depend on the concentration of each substrate and how much enzyme is there and the intrinsic activity or efficiency of that enzyme. So that relationship between the rate of product formation, or the enzyme velocity, and the substrate concentration is given by the Michaelis-Menten equation. So what we're measuring is velocity, or the rate at which product is made. So this would be like moles per second or moles per minute or what have you. An amount over a time is a rate. So that velocity, that's going to depend on the substrate concentration. And this velocity, it's measuring initial velocities. So before the rate of product formation before any equilibrium is even approached. Okay, this is like the initial enzyme velocity. Okay, so if we have different amounts of substrate, or if at different concentrations of substrate, the enzyme velocity is going to be determined by that substrate concentration, and then two constants, the maximum velocity, or the Vmax, and the Michaelis-Menten constant, or the Km. So this equation, if you've had a lot of math, you could recognize this as making a hyperbola if we plotted this out, right? And it's important to note that, let's see, how do we say this? Enzymes don't look at this equation and decide how they're going to act. Okay, this equation was derived by measuring how enzymes act. Is that clear? Okay. All right. Okay, so the two important constants that we'll look at are the Vmax. The formal definition of this is the enzyme velocity that you would get if you had a hypothetical infinite concentration of substrate.